Well, good afternoon. I'm excited to be here to share some information with this group on local field potentials. I'd like to give an acknowledgement to the support we've received for this seminar series from Alpha Omega. And thanks to Shelley for that great introduction to the Parkinson's Foundation resources. So the title of the talk will be, What are local field potentials? At the moment, that might be an alien concept, but hopefully by the end of this seminar, it will be a little more familiar. And we're going to talk about why they're important, specifically in the context of deep brain stimulation. And please feel free to jump in with any questions as we go through the talk. So these are some of my disclosures related to some of the work that I'll talk about today. So just to put this talk in context, if you've been faithfully attending all of the seminars to date, so far you heard from Dr. Aslam about what is deep brain stimulation. You heard from Dr. Baumgartner about whether or not you might be a candidate for deep brain stimulation. This is, again, focused specifically on Parkinson's disease. And then you heard from Dr. Stephen Ogeman about what your options are in the context of receiving deep brain stimulation surgery. And today, we'll talk about local field potentials and how these play a role across the spectrum of surgery and post-surgery in therapeutic management. And the final seminar in this session will be delivered by Dr. Drew Kern, talking about what's next. All right, so why am I here today talking to you about local field potentials? Well, in the context of deep brain stimulation, not only do we deliver stimulation for the therapy itself, but we can use electrophysiological information to help guide our surgery and also help guide therapy. So this is just a picture of the individuals that might be present during a deep brain stimulation surgery that you heard about from Dr. Stephen Ogeman's talk. And so obviously there's going to be a neurosurgeon present, there's going to be a neurologist, a movement disorder neurologist present, and then a neurophysiologist like myself will be available in the room. And what we'll be doing, I'll be helping coordinate with the neurosurgeon and neurologist to listen to the brain signals. This will be a conjunction of both the local field potentials that we'll learn about today, but also classically, we still use single neuron information to help guide our understanding of where we should place the deep brain stimulation electrode during surgery. So big picture, what we're going to talk about today for local field potentials and their role in deep brain stimulation, there'll be three main categories or three main ways in which they're important for this process. Number one, intraoperatively. So in either an awake or a sleep procedure. We can utilize electrophysiological information like local field potentials to help us determine if we're in the right location. And this can be done, as you'll see, we'll talk about single neuron information, but also we'll talk about local field potentials and we'll talk about specific frequencies. So here you can see I'm showing a heat map of beta power. Now, again, that's at the moment, don't worry about that too much. We'll get into the details of what I mean by beta power, but just keep in mind that we're using this information to help us find where X marks the spot in the brain. Then what we can do post-surgery is we can also use local field potentials from the implanted electrode to help guide initial programming and also optimize therapy. And this is a little more future forward, but also present day. So as of February of this year, there is at least one deep brain stimulation device that allows for adaptive stimulation. So using the local field potential information from an individual's brain to change its stimulation chronically. And so this is an important way in which local field potentials can play a role in post-operative therapy. So those are the big themes for today. And don't worry, this won't be too much of a deep dive science lesson, but because we are talking about electrophysiology or neurophysiology, we do have to talk briefly about the brain and what does it mean when we're looking at these signals. So here you're looking at a picture of some of the important brain areas for deep brain stimulation in Parkinson's disease. So we're targeting these parts of the brain that are important for movement and that are dysfunctional in Parkinson's disease. And really the thing I want you to take away from these big colorful polygons and connected lines is that these brain regions are all communicating with each other via neurons. So you can have inhibitory or excitatory neurons, and neurons are just special cells that make up your brain that deliver electrical patterns that drive changes throughout your brain. And so so each of these big structures, the cortex, these parts of the globus pallidus, and importantly, the subthalamic nucleus, which we'll talk a lot about today. This is one of the targets that we know a lot about in terms of local field potential information, are made up of these individual neurons that we are changing their pattern with deep brain stimulation. So the main two things you need to keep in mind for today's talk is that these special cells in the brain, neurons, communicate through electrical impulses. And these impulses create measurable electrical signals in different brain regions. And so that's what we're using when we record either single neuron activity or local field potentials. We're recording the actual activity of cells in your brain or in that person's brain. Okay, so I'm focusing on local field potentials, but there's also a way to record global field potentials. So we're going to zoom out, looking at the whole brain. What does it mean to say we're recording from global regions of the brain? Well, imagine these are just electrodes that we placed on the surface of the skull or surface of the brain, but you can also imagine EEG, which sits on the scalp. And these allow you to record signals from the surface of the brain or the surface of the scalp. And this usually covers a large territory of the brain. So it doesn't allow you to get local information. But importantly, we can't just look at this raw trace. So this is actually a raw electrical signal that we're recording from these global locations. But what you've probably heard about in your maybe some courses you've taken or in the public or in the popular news, we can convert this signal into distinct waves, distinct waveforms. And that just means we're looking at how fast these waves are cycling. So we can have very slow waves called delta waves. These are cycling in 0.5 to 4 times per second. We have theta waves, alpha waves, and beta waves, and gamma waves. And these are just different ways of chopping up the speed of these waves that have been isolated from this single global signal. And things you might be familiar with, this is ways in which we can record from larger parts of the brain is called electroencephalography or EEG, which we've been doing for over 100 years. Electrocorticography, where we're recording from the surface of the brain, or fMRI. These are lots of ways to get this kind of wave-based information from larger parts or larger regions of the brain. But what about when we want to get local? Zoom in on some specific part. So let's see if we zoom into the brain. We're going to target one of those important deep brain structures. So we're going to go into the subthalamic nucleus, this red circle. And all we've done is we've inserted a recording electrode deep within the brain. And so that we're recording a very small region of tissue. In this case, it can be, this is showing 100 microns, but it's usually between 100 microns to 1 millimeter of tissue. And these are many, many neurons that are close together, all changing their electrical activity, but you're still getting a single raw trace, but it's giving you information about a very, very localized region in the brain. So this is what we mean by local field potential. And so to give you that in context, so here, if we're looking at EEG, this is recording on the surface of the scalp. It's covering a larger part of brain territory. You get closer to the surface of the brain, still kind of a larger part, 0.5 centimeters. But as soon as you get into the local field potential, you're recording from a much smaller piece of tissue. So you're getting more localized information about what's happening in the brain. So again, we can, same thing as with the EEG or fMRI, we can convert those local field potentials into individual waveforms or frequencies. And so here I've highlighted the beta frequency, which is a moderately fast wave from 12 to 30 hertz. And we know from many, many years of research, probably over 15 years of research, that this specific frequency range can exhibit pathological changes in Parkinson's disease. And it's something that we can use to target, to modify for clinical benefit. So just to make this clear, I want to show two conversions of how we get from that raw voltage trace that we're recording from a localized part of the brain. That first step is to convert this trace into frequency bands. This is important. It's our first step, because we want to know what's happening in each of these frequency bands, since we know that a particular frequency band is important to us for understanding how to target the brain for deep brain stimulation. But there's many plots that I'm going to show you today, and many plots that or many figures or visualizations that you might see in the clinic that I want to help you understand and interpret. So right now we have our raw trace. We've converted that raw trace into different waveforms or frequency bands. There's one more thing we can do. We can actually try to represent the relative power of these frequency bands. So we might imagine that for that raw trace, we had different levels of power in these frequency bands. So in this case, beta might be higher than theta. And what we can do is then we can measure those relative differences in a line plot. And so this is commonly how we try to understand at any given moment what's happening in the brain when we're trying to make decisions in the operating room or in the clinic. And I'll show you one quick example. This is a plot of a power spectral density plot that we might look at that tells us about local field potentials. So just like I'm showing here, we have power on the y-axis, we have frequency on the x-axis, and we can see that when stimulation is off or deep brain stimulation is off, the medication, the Parkinson's disease medications are not being taken, that we have a higher beta power. So beta is higher, the power is higher in this frequency range. So that's how we got to this power spectrum. We went from the raw trace to the frequencies, and then we compared those relative frequencies in their power. So for the rest of the talk, I'm going to show you many, many different pictures like this. I want to make it clear how we got from our localized recording to this information. All right, so what we're going to talk about now are those separate categories. So we're going to start off with how are LFPs helpful in intraoperative surgery for deep brain stimulation. So one of the other methods that we can use for electrophysiology are single neuron recordings. So these are the typical brain regions that we cover or traverse when we're trying to get to the subthalamic nucleus for deep brain stimulation therapy. And you can see that in these traces, again, a raw voltage trace, you'll notice these single individual spikes that come off of that main dense trace. These are what are called action potentials of an individual cell or multiple individual cells. And so we're seeing its individual unit activity. You can see as we go to different brain regions, that activity actually changes in the way it looks. And this has been historically very helpful in trying to figure out where we are in the brain when we're performing these procedures on patients. So just so you can hear what we hear in the operating room, this is an example of surgery. Let me make sure the volume's up here. So I think it's very evident that you can both see and hear there are really distinct differences when we're looking at the single neuron activity as we cover these different brain regions. And importantly, we're really trying to find that SDN, that's where we want to place the deep brain stimulation electrodes. But also we always look for the substantia nigra pars reticulata, which is just below the SDN, to make sure we've got the true border. But this information can vary from patient to patient. And so sometimes it can be helpful to use local field potentials to help determine where the best spot in the brain is. Oh, sorry, it sounds like you couldn't hear those. Well, hopefully you could at least see the differences in the way they looked. Thank you for that feedback. So you want to think about, in the context of recording from these different brain regions, if you're recording from a single neuron, it's as though you've placed a microphone, if you're thinking about a stadium in which people are there to watch one of the March Madness games, and you have a microphone hovering above the stadium, and so you lower that microphone all the way down to a single individual participant, and you're recording just from that one person. So that's essentially what it's like from recording from a single neuron. But what about if you raise the microphone up above the stadium, or a portion of the stadium, let's say half the stadium that's supporting one particular team, you're now getting closer to the average activity of many participants. And that's closer to the way we think about a local field potential. And again, this allows us to extract these brain waves, look at the beta range, and then try to understand how that beta range is involved in movement, medication, and stimulation. That's what we'll talk about next. So just to show you an example of how this might work or how this looks compared to those single neuron recordings. So again, as we're traversing those different brain regions in the basal ganglia, we get different individual neurons as we get to SDN. And here's showing one of those power spectral density plots. In this case, instead of a line, we're showing color to represent power. And you can see as we get closer to the subthalamic nucleus, there's an increase in that heat or increase in the color that's telling us that there's more of that beta frequency. It's higher in power, telling us we might be close to a location in the subthalamic nucleus that's a hot spot for where we want to target our deep brain stimulation electrode. And then when we compare using that spectral plot, when we compare local field potentials between the subthalamic nucleus, the thalamus, and the STN, we can see that we're able to differentiate those structures similar to the way in which we could do that in the single neuron recordings that we saw earlier. So local field potentials are certainly helpful in differentiating these brain regions. There was some earlier work that also indicated that not only could you potentially tell where STN was located, but also that you could find the hotspot in STN where you should consider placing the deep brain stimulation electrode. This is a publication that over 300 recordings found that in the top part of STN, which we call the dorsal STN, you can see here, these are many, many individual recordings stacked on top of each other. The red is indicating that the beta here on the x-axis from 13 to 30 hertz is pretty high. It's a higher end of power compared to the bottom part of STN, which is has much less red and much less yellow, indicating it's lower in beta power. And so this was an indication that if we wanted to find that hotspot for beta, it was very likely across many, many subjects to be located in the top part of STN based on that beta LFP power. Another later study actually used the deep brain stimulation electrode as their way of monitoring where they were in the brain during the surgery. So this is showing a picture of the 4 recording contacts from the deep brain stimulation electrode. Here you're seeing above the power spectral density plots. And right now you can see that there's not very much difference between the power and these different frequencies. But as they move into the subthalamic nucleus, you can see that red electrode, once it enters the top, has an increase in power in the beta range. And then they show that as you further move into that dorsal portion, the green continues to increase, and now you're seeing an increase in the yellow, indicating that contact is now in the hot spot for beta. And as you can see, it's now taken over the location for yellow. Green is now either equivalent or coming down. And now yellow, as it moves into that position, is also showing the increased beta. And then same with red, just showing that they were able to demonstrate that recording with the deep brain stimulation electrode alone was able to help them target based on the beta power information. And so they could then localize exactly where they wanted to place the electrode using that information. And in this case, this is an electrode that has not only just 4 levels. So here they made sure that their beta hotspot was placed with the contact 2, but they also made sure to get the second highest beta with that second contact. So they were using this information to identify that beta peak to determine where to place the electrode. In addition, in the operating room, we are able to use a commercial system that can tell us where the increased beta is. So in this case, we can see that for a particular depth location, we're getting a nice beta peak. And then we can implant the deep brain stimulation electrode and then do an additional recording. And then from that information, just like I showed you in the last plot, using the LFP beta power based on those 4 contacts, determine which contact might be optimal for therapy, or if we think we might need to move the electrode based on this information, we can do so using the LFP power in beta. And this is all based on the idea that you can both identify this beta hotspot that you're seeing in yellow, but also you can hypothesize where your stimulation, which is shown in red, might cover that beta territory. I mean, this is the ideal scenario where you are able to cover the area of that beta hotspot with your stimulation from the electrode. So moving on to how we might use this information, use the local field potential information post-operatively for initial programming or optimizing therapy. So now we're not just recording from the electrode as it's moving, we're recording from the implanted electrode post-surgery. So the patient is now in the clinic coming for their initial programming. So there's several things that we've learned about using the beta frequency specifically in the subthalamic nucleus. One thing that several different studies have demonstrated is that you can get an enhanced beta activity. There's enhanced beta activity during rest, but suppression during movement. As you can see here in this power spectrum plot, in the red is during movement, which is suppressed compared to resting. With regard to Parkinson's disease medication, you get a decrease in the beta power, that's shown in orange, when a patient takes their medication, their L-DOPA medication. When they're off, you can see an increase in the beta power. And then finally, there have been several studies showing that you can observe the efficacy of deep brain stimulation based on the change in local field potential power and beta. So here in this plot, we're seeing in stimulation off in red, both of these are off stimulation. And then when you turn on, when you get best clinical stimulation is in black, so you get a suppression or reduction in the beta power. But also interestingly, as you increase stimulation stepwise, almost like a drug dose response, you see a stepwise decrease in beta power. So these are the three ways in which we have been able to utilize this beta LFP signal to help inform pathology and movement, therapeutic response in terms of medication, and therapeutic response in the context of deep brain stimulation. So these things motivate how we might utilize this information in the context of programming. So there's also, as I mentioned, this idea of an anatomical sweet spot. So this is data that's been accumulating over several years across several different studies, showing that across subjects, when you stimulate in a particular region that exhibits increased beta, you have a greater likelihood of inducing an optimal response. And that's thought to arise in this dorsal posterior lateral portion. And so this is the location that we try to target for surgery, but also in newer DBS electrodes, you can change the direction of your stimulation to try to hit this beta location. And again, I showed you this image earlier, but the idea is to try to cover that beta hotspot with your stimulation. And what we'll look at later is that in the clinic, we can now track the LFP beta power as we deliver different levels of stimulation and observe in real time, as you can see in blue, the beta power decreases as stimulation increases in orange. So one way in which potentially local field potentials could help in initial programming is reducing the amount of time it takes to optimize therapy. So in the first few visits, the clinician's goal is to get the stimulation parameters optimized to the best they can be for that patient's specific motor symptoms. And the goal is really to maximize the DBS benefit while also minimizing the adverse effects. So finding that optimal therapeutic window. So there are several parameters that the physician can modify for deep brain stimulation. They can change the rate of stimulation, the amplitude, and the pulse width. And with newer electrode models that most of the companies now have, you can also change the direction of stimulation, the fractionation of stimulation across contacts, contact number, and also you can record local field potentials to help guide programming. So this is great. This has been an increase in DBS technology, but also it's led to an increase in the time it takes to figure out which of these features are best for each patient. And in particular, with the increased number of contacts, the ability to change the direction of stimulation, and the ability to change the fractionation, so how much you're stimulating on one contact versus another, allows for the physician to create these complex stimulation shapes. And so the question is, how quickly can we help that clinician get to this complex, unique shape? So to briefly review, what does the program process look like now and how can LFPs help? So this is a typical DBS electrode with 4 levels. The 2 middle levels have 3 different segments that are arrayed around the circumference of the electrode. And what happens in one of the first visits, once you have your deep brain stimulation electrode implanted, is the clinician will conduct what's called a monopolar review. So they'll, at each contact, they'll increase the stimulation until they identify where the subject either experiences maximal benefit or an adverse effect. So in this case, they were plateauing at about 75% improvement in rigidity until they received an adverse event for this particular contact. So the clinician will do this for all possible contacts. And with these modern electrodes, this can mean several different tests to figure out which of those, in this case, these 2 segments, which allowed for maximal improvement with minimal adverse effect, are ideal. So this can take several visits and require lots of trial and error. So the question is, can we use local field potentials to improve this process? In this case, there is a commercial platform that will allow you to observe the local field potential information in a power spectral plot that you now know how to interpret. That's showing us what that power looks like in the beta frequency range when we're comparing contact 1 to contact 3. So we have a very high beta peak here and between those 2 contacts at 20 hertz. So prior to about a month ago, these platforms only allowed you to look at bipolar information, so you could compare across different levels of the DBS electrode. And you could also compare between segments, those 2 middle levels that allowed for you to look at those individual segments. But you'd also have to interpret where did that highest beta power peak come from. In this case, we can see that it's coming from contact 0-2, which is our highest beta peak. But it still wouldn't tell you exactly where to stimulate, which potential location would be optimal. And so we conducted a study to determine if we could record from a single contact using a commercial platform. Could that single contact information predict or provide information to the clinician about where to start their therapeutic stimulation? So here I'm showing you an example of recording from an individual contact from the levels. This is, again, the power spectral information. You can see very clearly in the levels and in the segments that there is one specific contact that has the highest beta. And the question is, if we know that these 2 locations have the highest beta, if the clinician chooses to stimulate off this contact, does it provide good therapeutic benefit? So can they skip that monopolar review that I showed you earlier and just select this particular contact? So we conducted a study to assess that very question. So we had 10 subjects in their initial programming session. They had to come in off medication, and we compared this single contact LFP recording to the traditional monopolar review. And in a double-blind process, we compared the two different contact outcomes in terms of their clinical benefit. So what we found is when we compared the traditional monopolar review to the single contact LFP identified location, that there was no difference in the outcome, showing that using the LFP alone was equivalent across their clinical benefit. This was true for the subscores of rigidity, bradykinesia, and tremor. But importantly, it also saved a tremendous amount of time. So the initial monopolar review that I showed you is commonly conducted to identify the best location, takes on average about 124 minutes, whereas just recording that single LFP information in conjunction with a single monopolar review for that contact takes about 16 minutes. So the clinician was able to identify much faster which contact to start stimulating from based on the beta LFP information. And now this, as of March, this information is now available in the clinical platform, in the commercial platform. And so the clinician can now see where that single LFP contact is highlighting the maximum beta. In this case, it's showing that it's on contact 2. So now they can start programming at that contact using this LFP information. So finally, another way in which LFPs might be helpful in the context of deep brain stimulation therapy for Parkinson's disease is in the chronic context. So I'm going to talk briefly about how we can use this LFP information to help us better understand how stimulation is affecting daily living activities, but also potentially now with the FDA approval of adaptive stimulation, how this new feature might also improve therapy using local field potentials. So from a chronic LFP standpoint, we're hoping that this information can give us better insights in the brain state over time. In particular, can it help inform sleep activity patterns, give us better insight on on-off fluctuations, and also give us better understanding of the interaction between medication and stimulation. So again, we'll start from the clinical platform or commercial platform that allows that clinician to identify a specific LFP peak. In this case, we can see in this individual that they had a very high peak in their STN at 18.5 hertz. So the clinician might choose to track this particular LFP peak chronically. Then we can ask, what does this peak do if we track it over 2 weeks during a typical daily activity pattern of an individual? So in this case, these subjects were given a device that tracked their activity, their sleep-wake activity, and we're also tracking that LFP beta peak. So here is their activity patterns. You could see over a 2-week period, they have a typical circadian phase where there's a lot of activity during the day, low activity during the night. And the question is, what happens to that beta LFP? And so we can see that there's actually a very intriguing circadian pattern to that local field potential information. It does have some changes in amplitude in the first 3 days. It increases over the next 12 days. And if we were to dive into this information, we might also see subtle changes related to medication regimen. But these are some of the insights that we're hoping to take advantage of with the chronic information that LFP can provide. When we look across patients, when we track their LFP power over awake and asleep, we actually find that across all patients, their beta power is significantly decreased in the night compared to the day, indicating another potential role for LFP beta in differentiating sleep versus wake in the circadian rhythm. And finally, now that in one commercial platform, adaptive deep brain stimulation is now available, the idea is, in this case, as I've shown you, the system can record raw LFP using a beta power signal, the clinician can now set a threshold that once that threshold is exceeded, stimulation will be turned on. And when that threshold is not exceeded, it might reduce stimulation. So you can increase and reduce stimulation on a moment-by-moment basis to help mitigate the fluctuations the patient might have been experiencing, particularly when they take their medication. And so this is showing a reflection of how this the beta power is actually either crossing that threshold or not crossing that threshold, and how it's changing the stimulation gradually to address those threshold crossings based on LFP beta power. So again, this is very new technology. We're still, the field itself is still understanding how best to utilize it. It was only FDA approved as of February. First subjects were initiated in this programming in March of this year. But the hope is that this will improve the interactions between stimulation and medication to help reduce the potential overstimulation that might occur when the patient initially takes their medication, but also increase on time and reduce off time. And also potentially manage dyskinesias, again, related to the interaction between medication and stimulation. So we've covered lots of territory today. We've talked about how LFPs might be important for intraoperative investigation, helping targeting the optimal deep brain stimulation location, how we might potentially use local field potentials in the programming process, particularly to make it more efficient and get to that optimal location, and also in the future, or even present now, near future, using this information to help us understand how these brain signals are changing in someone's daily activities, but also utilizing it for adaptive deep brain stimulation. So I'll show you how, in general, the LFPs have augmented what we currently do, and also not necessarily replace, but augment how we normally target deep brain, or how we plan for deep brain stimulation surgery. So we certainly use imaging to identify our location of interest. We commonly use single neuron activity, as I showed you earlier. And then there's a programming process once the patient's implanted. So I've shown you how, in the context of surgery, we can utilize that beta hotspot as a part of our planning process, but also we can utilize intraoperative beta LFP to augment or enhance that single neuron activity. In the context of programming, we can use the LFP beta again, bringing all these pieces together to help guide programming. So again, thinking about this workflow, we start with imaging, and this is something you've likely heard from Dr. Ojuman. We have our electrophysiology, both single neuron and LFP, our kinesthetic testing, our macrostim testing, intraoperative imaging, but this is also in conjunction with using our LFP-informed information from a recording system, which helps guide where we implant our electrode. And then we can use LFP confirmation once we've implanted the electrode. Once they're in the clinic and we've identified where to potentially start looking for that LFP power, we can do a monopolar review, identify the contact of interest, and then deliver that stimulation titration. So here again, you're seeing, once we've identified this contact with the maximum beta, we record a peak beta and then stimulate in increasing increments of activity to see if we can suppress that beta power, identifying that we're in the right location. And then we can do post-operative reconstruction to determine exactly where those contacts were located and measure the volume of tissue activation to see if we're targeting that hotspot. So we've covered today how LFPs are increasingly being used to augment DBS targeting in the OR, how many of these options are also available in the operating room, but also in the clinic to help us bridge the gap between what we observe in the operating room and carry it forward into the clinic to help deliver ongoing therapeutic insights. And also now in this current future, we can potentially utilize these LFP signals to modify brain activity using adaptive stimulation. So thank you, and we'll be happy to take any questions.